Okay, hello everybody. Thank you very much, Allison, for the invitation to speak here today. I'm very excited to tell you about some of our unpublished work. I'm an assistant professor at UC Berkeley, and in general, my lab is interested in understanding how mutations associated with neurodevelopmental disorders affect the function of neurons and how this change in neuronal function affects brain circuits and brain activity. And we have a particular interest in autism and epilepsy-associated disorders. So today I'd like to tell you one story of our work using human cortical organoid model to look at one particular disease that we've been interested in. And this disorder is called tuberous sclerosis complex, or TSC. And this is a multi-system developmental disorder. It causes the formation of benign tumors in multiple organs. However, the neurological or neurobiological implications of this disease are the most detrimental to the patients. So patients with this disease have a very high prevalence of epilepsy. This is a very severe form of epilepsy that can start in infancy and often becomes intractable. The patients have varying degrees of intellectual disability and cognitive dysfunction. There's a high prevalence of autism, and so this is a syndromic autism spectrum disorder. There's other psychiatric conditions as well. And pathologically, this disease is often identified by the presence of these so-called cortical tubers, where the disease gets a name from. And these are macroscopic regions in the cortex of highly dysmorphic, disorganized neurons and glia. And so these are often an example of a focal cortical dysplasia, where there is these discrete regions where you have these developmental malformations. And at the molecular level, we know a lot about what causes this disease. It's caused by loss of function mutations in either the TSC1 or TSC2 genes. And the products of these genes are critical negative regulators of the MTOR signaling pathway. So when you have mutations in TSC1 or TSC2, you lose this negative regulation of MTOR. MTOR signaling becomes chronically activated and deregulated. And we can see that by looking at the phosphorylation state of the downstream targets of MTOR. And in my lab, we're interested in understanding how this MTOR pathway and deregulation of it affect neural development, cell morphology, neuronal communication, and excitability. But for the purposes of today's talk, I'll tell you about how these mutations in the MTOR signaling pathway is important for human neuronal development and function. And the reason we work on both mouse models and human cells in the lab, and the reason we have moved to a human system is because these tubers actually don't form in mouse models of the disease. You see some phenotypes recapitulated, but these cortical tubers seem to be kind of a human brain-specific phenomenon. And as Allison alluded to, that may be because mouse and human brain development is quite different, particularly cortical development. So here's an image, a section of a mouse brain, and you can see the cortex is this thin little stripe on top, in comparison to the human brain, where the cortex is this highly enlarged and elaborate structure. And so the cortex in particular has a unique developmental profile in humans. And that is a result of different types of neural progenitor cells that are found only in humans and not in rodents or other species. And this gives rise to unique proliferative zones that, again, are present only in humans and not in mice. So if we want to understand a human neurodevelopmental disorder that affects the cortex, we really need a human system, and preferably a human organoid system, to recapitulate this. And so we took an approach that you're all familiar with, where we can start with human stem cells, and we have either iPS cells derived from TSC patients, or we've made a lot of cell lines using Cas9, where we can just engineer mutations in TSC1 or TSC2 and differentiate these into neural progenitors, neurons, and these cortical organoids, which is what I'll focus on today. And I'll just say we've made a large panel of cell lines, and in the interest of time, I'll just show you a little bit of data looking at cell lines where we have homozygous mutations in these TSC genes. Okay, so here is the organoid differentiation protocol that we're using. It was originally developed by Sergio Pasca at Stanford. And basically, you take these stem cells and you detach them from the plates so that they become 3D structures, and you go through a series of steps where you can generate first neural progenitors to expand these organoids, and then you induce neuronal differentiation, and then sometime later you start to produce astrocytes. And this kind of the timing of this development pretty closely mimics human cortical development, so we think that this is a good model for looking at this disease. And so we were first interested to understand how mTOR signaling pathway behaves during this differentiation process. And what we found is that rather strikingly, we see a very strong suppression of mTOR signaling, and we see that by looking at the phosphorylation state of these mTOR targets. We see a very strong suppression of mTOR signaling as these cells go from being proliferating neuroprogenitor cells to becoming differentiated neurons right here around day 30 or so. And if we take one of our organoids that has a homozygous mutation in TSC2, we can see that this suppression of mTOR signaling or this regulation is actually lost. And so these cells maintain very high levels of mTOR signaling throughout neuronal differentiation, basically as high as levels as you see in stem cells, in which mTOR signaling is almost at a ceiling. And so loss of this gene causes a failure to shut down mTOR signaling during neuronal differentiation. And so what effect does this have on neurogenesis? What we found is that this chronic high level of mTOR signaling in these organoids actually prevents neurogenesis. So if you look at a neuronal marker here, MAP2, and in black are wild-type organoids, you can see this starts to come around day 50. And we can see in organoids where TSC1 or TSC2 have been knocked out, the emergence of these neuronal markers is reduced and delayed. Okay, so when you have high mTOR signaling, you actually suppress neurogenesis. And we also see that if we look at a different neuronal marker at day 100, it's reduced in these knockout organoids. By contrast, if we look at an astrocyte or glial markers, we can see that these are actually enhanced. And so here in blue and red are these knockout organoids that actually produce very large numbers of astrocytes and seem to do this at an earlier stage than they normally would. If we just look at the cells in these organoids and we stain for neuronal marker MAP2, what I hope you can appreciate here is a TSC1 knockout organoid and a TSC2. These cells, or the neurons that do develop, are highly dysmorphic and enlarged, and they have very high levels of mTOR signaling seen by the phosphorylation of S6. And interestingly, we don't see these changes in cells from heterozygous organoids. So this data suggested that this failure to repress mTOR signaling during differentiation is actually problematic for neuronal development and is shifting the balance between neurogenesis and gliogenesis. But one thing I didn't mention is that the TSC patients are actually heterozygous for these mutations. And as you can see here, we didn't see any phenotypes in our heterozygous organoids. And this may be because these tubers actually so although the brain of these patients are heterozygous, the prevailing model is that these tubers might actually form from a second hit mutation analogous to how some tumors form. And so this model, called the second hit model of cortical tuber formation, suggests that at some point during neuroprogenitor proliferation and expansion, some subset of cells acquire a second hit mutation that causes biallelic inactivation of TSC1 or TSC2. And that these cells can proliferate, but when they have to migrate out to form this structured layer in the cortex and differentiate into neurons and glia, they run into problems, and so they form these clonal kind of regions of cortex with these highly dysmorphic and dysplasic cells. So this is a model in the field for how you get these focal regions of dysplasic cortex, but it's been really difficult to test this hypothesis because, again, the mouse models don't show these, and it's difficult to get human tissue to do the sequencing to confirm this. So we thought we could use the organoid model to actually formally test this possibility. And so what we did is we engineered ES cells to have a null allele for TSC2. And on the second allele, we generated a conditional loss of function mutation that was Cre-inducible. So these organoids are heterozygous for TSC2. Unless you add Cre, then they become homozygous knockout. And also, we also use Cas9 to engineer a Cre reporter in these same organoids so that when any cell that was transduced with Cre will turn red, and its progeny will turn red, so we can follow lineage trace and follow the fate of these mutant cells over time. And so just to show you that the system works, if we add Cre to these cells, we get complete loss of TSC2, and we get very strong upregulation of mTOR signaling. And if we add Cre in a very sparse way to mimic a sporadic mutation, where we can look at these cells in red that have the full mutation, they have complete loss of TSC2, and we can use GFP to look at the neighboring heterozygous cells, and we can see that these cells have cell autonomously very high mTOR signaling and show this very dramatic enlargement. And so we took this approach in the organoids, and I hope you can see this. I apologize, it's a little bit hard to see. So here's an organoid here. And in red are these so-called tuber-like regions that we've generated by adding sparse amounts of Cre virus. And here in red are these knockout cells within these tuber regions. And here's what they look like at day 50. And if we monitor them over time, you can see that these cells become dramatically enlarged and very dysplasic and dysmorphic, and they form these kind of focal tuber-like regions. And so we think that this could be a very good model for tuber formation in TSC. And just to show you a side-by-side comparison, here is a human section from human fetal cortex at 23 weeks of gestation. And you can see this is the early tuber that is forming in the cortex. And you have these very enlarged balloon-like cells that have high mTOR signaling. And here's one of our cortical spheroids. And hopefully you can see that these cells look very similar, and they have this similar reactivation of mTOR. And so we think this is going to be a good model for studying how these tubers and these tuber cells form in early development. And if we look at characterize these tuber cells, so here you can see these very dysmorphic cells stained with TD tomato. They express very high levels of neurofilament proteins. This is something that's characteristic of the patient tubers as well. And when we go back to look at this neuron versus glia fate decision, we can see, again, that this cell-autonomous loss of TSC2 leads to a preferential differentiation into astrocytes at the expense of neurons. And so what we've done here is these red TD tomato cells are the knockout cells, and then the neighboring heterozygous cells we have labeled in green. And so when we can quantify whether these cells have produced neurons or glia at a certain stage of development, and what you can see here is that these knockout cells are generating many, many, many more astrocytes and much fewer neurons than their neighboring heterozygous cells. And so the next step was to test whether this phenotype could be preventable or possibly even reversible, and also test that this is indeed due to mTOR overactivation in these cells. So we used the drug Rapamycin, which is a very potent and selective inhibitor of mTOR, and we applied Rapamycin at the same time that we applied Cre to suppress mTOR signaling during the differentiation process. And what I hope you can see is we very dramatically prevented these tuber cell formation, and we fundamentally switched the cell fate decisions of these TSC2 knockout cells. So whereas they were generating high numbers of astrocytes, in the presence of rapamycin, these TSC2 knockout cells actually generate large numbers of neurons. And interestingly, even in the neighboring heterozygous cells where we otherwise don't see phenotype, we see normal development, rapamycin actually promotes neurogenesis in these cells as well. And so this was somewhat of an unexpected finding and suggests that mTOR signaling is actually fundamentally involved in the differentiation process and the decision about when and whether to become a neuron or to become an astrocyte. And when you have deregulation of this mTOR signaling, as you do in TSC, this affects cell fate decisions, and you end up with an abundance of astrocytes that are enlarged and dysmorphic and cause problems to the surrounding neurons. Okay, so just to summarize what we found is we found that these mutations in TSC1 or TSC2, which are causative for tuberous sclerosis complex, strongly impact the development of neuron astrocytes, and we did this using this cortical organoid model. And we found that normally you need to suppress mTOR signaling during neurogenesis, and that the failure to suppress this is detrimental. And we found that homozygous loss of these genes is actually required, so we didn't see many phenotypes in the heterozygous cells at this level. And we do find support for this so-called second hit model of cortical tuber formation, that you really need both copies of the gene to be disrupted in order to get these tuber-like cells. And this developmental abnormality can be prevented by early rapamycin treatment. One thing I didn't mention is we tested whether later rapamycin treatment could reverse the phenotypes, and we see kind of a partial rescue. And we're in the process of testing kind of different developmental windows of treatment to find out when there might be a critical period for normal formation and normal cortical development. So I'll end there and thank my lab, and I just want to say that this project is a close collaboration with our colleague Derek Hockemeyer, and I thank our funding, and I'm happy to take questions.
Questions for Helen? Just wait a moment.
Hello. I was wondering, for the cell fate change that you saw in the increased astrocyte number, what do you think is happening with the astrocytes? Do you think that they are maybe contributing to epileptic phenotypes? And have you looked into that?
Yeah, that's a great question. So I should have said that this phenotype of having an abundance of astrocytes recapitulates what you see in human patient tubers as well, that you also see expression of a lot of astrocytic proteins, and there's astrocytes there. And yes, it's interesting to think about how this abundance of these dysmorphic reactive astrocytes might be affecting the surrounding neurons. And certainly these tuber regions are often epileptic foci, and they often are surgically removed in cases where the epilepsy is intractable. And so the next step that we would like to do is understand how these kind of tuber-like cells are affecting the activity and the network properties of the surrounding neurons. That's a great question.
Yeah, I was just wondering, it's really interesting to get more neurons. So I was wondering whether those neurons persist or not during development.
Yeah, so we've only looked at one time point so far, which is around day 100. We haven't let the organoids grow much longer to see if they persist, but I would imagine that they do. So yeah, we're interested now in investigating kind of the mechanism for this, and we think that MTOR signaling may be directly regulating the transcription of gliogenic genes, and that you need to suppress that transcriptional activity during neurogenesis. And so if you have a stronger suppression of that, you promote neurogenesis.
Jake.
There's some interaction between the TORC1 and TORC2 complex, and the tuberous sclerosis is regulating TORC1 only, but TORC2 is interesting because it has effects on cytoskeletal activity and reorganization and cell survival. Do you see some crosstalk between the two things in this?
Yeah, so we have projects in the lab that are directly looking at the involvement of mTORC1 and mTORC2 in the context of TSC. Because there is some crosstalk there. And what we often see when you have mutations in the TSC genes is that you have this hyperactivation of mTORC1, but you also have a suppression of mTORC2 activity. So I don't know if you saw on one of the Western blots, there's really strongly reduced phosphorylation of AKT, for example, which is the mTORC2 site. And so yes, we're interested in the possibility that some phenotypes of TSC are actually due to this suppression of mTORC2. And trying to tease that out, because rapamycin suppresses mTORC1, but it also indirectly suppresses mTORC2, so you wouldn't be fixing those phenotypes. And so we're trying to figure out ways to rebalance mTOR signaling to get kind of normalized activity of both complexes.
I do have a question. Yeah. So the proof of principle that the two-hit hypothesis actually happens in autism is very nicely demonstrated by your model. However, I think most of people always think that the second hit will not be in the same gene, but something else, or even an environmental insult that might lead to the clinical manifestation. So now, I mean, you are proposing the two hits in the same gene. I mean, how do you see that?
Yeah, I think that in TSC, it's a specific case where, so in the tumors that you have in peripheral organs, people have sequenced them, and you do see evidence for biallelic inactivation. So you need the second hit in the same gene as you had the original mutation. So there it's pretty clear. So we think that is also happening in the brain in these tubers. However, I imagine that's kind of specific to TSC, and that for these other disorders where you have kind of a second hit, it could be in a different gene. It could be an environmental insult. So in TSC, there's studies in mice showing that if you're heterozygous for TSC mutation and you have an additional environmental insult, that is what you need to get any behavioral phenotype. So I think, yes, the second hit could be something outside of the original gene in many cases.
Do you know, if I missed that, what is the mechanism of mTOR suppression during the onset of neurogenesis? Do you know what causes it?
We don't, actually. So that's something that we're pursuing right now to understand how it's being regulated. We don't know the mechanisms yet, and then we also would like to know, in turn, what mTOR is regulating to control these cell fate decisions. Yeah.
I think we should stop right now. Thank you again, Helen. And